figured this out by now, but I want to make sure that everybody understands one thing.
Page issue that deja haku sasha simbanaya, Samagi haku chetela yes.
There are two strips of paper that have no capital letters on them.
That and the inchigi igichiwachi or capital letter igichiwati mechugi shogumas na shoganidus.
One of them has an arrow above it, and the other does not have an arrow above it. The one that does not have the arrow is the original. Sorry. Look closely at the one that has the arrow above it, and you will be able to notice the change when you compare it to the original. I'm going to give folks 5 more minutes to order them from the most similar to the original to the most dissimilar from the original, and then we will discuss.
Tatanda yang tando kamangas players kamanga green laya and chisu pangana stukure sena pisichi orwa tomachi ninjur machin big pisichi orwa de laya and in dachi shiro and dashi sukura that they don't tell ni shiti karimba tampone chene kolaya and a cheper chishu sade and the tamatela to stitch panduroches that the pantigala kamanga players.
So 5 more minutes to feel good about your order, and then we'll move on.
Do
you all need more time, or you got it? You're finished? Okay. Y'all are are you still working or done? Oh, maybe they're all done. Are you still working or done?
Yeah, we're done.
Done? Okay. Look like they're
yeah, some are still working.
Okay.
Yeah.
Okay.
Similarity with the original.
Yeah. So this is a formative assessment, so I'm not going to give you the answer.
Okay.
Yeah. I'm just trying to see what you know. Should I get on my phone too and hold them all in here?
I think so.
Okay, if you are not done, that's okay. This is designed to have 2 parts. 1 is you order it now. 2, we review the different types of mutation on the screen, and then you can make corrections if you want.
Tanda kari chombade tagana duba ches, dela yang asuge, rumba ni gana duga yens, ta tampo nga suge tanda de ninjur ge pangram te de rim shin te pangra de nga sunyam te chita ge ins ro kar de yara gangla ten de ta ge ins te la tanu ge je la ya ni che su ge ya su su te la ya jura tang ko na te la ten de tan jura tang na degrees.
So let's go over the different types of mutation.
Da tanda ninjur ge re te ta la ya chita ge ins.
Before I do it, the one thing that I wanted to build from that we did yesterday is there are two ways to think about mutations.
Da kesang asu ge ngoto chi batti gang la ya khanun chi ate la ya lenga su ge ninjur ge ko la samlu tang bi kaps ani samlu tang tang man ro ani samlu tang na degrees.
The first way is to think about what happens in the DNA code itself.
Chi ka re system la ya DNA ge sanda chi warwa tig nang la ya jira ka ri chi chong du se te ta yo res.
The second is what is the impact on the polypeptide chain.
Nibati ka re system la ya til te ni de de nga su ge tsi gi thwa wa tsi gang la ya ni shuchen ka ri then du te ta yo res.
I'm going to share these with you from the DNA code perspective, and then we'll talk about how that impacts the polypeptide chain, which is what Miguel talked about yesterday.
Da tangpo de la nga su ge tangpo de la ya ani DNA gi khurang ga sanda ngola ka ne do chi ba de che ni ti je la ya ti la te ne de nga su ge tsi gi thwa gang la ya ani khesang geng Miguel la gi song yo wa ti gi gang la shichen ka de the me te ta ins.
So, let's just start with normal replication of DNA. We have the original code here, and then we have the copied code down here. Replication happened smoothly, there were no changes.
Da de bana ka su kana do ge me te ta ge da de ka su ro da de ka su tangpo de la ya nga su ge tangpo thomi ge khorum ti che tila te de ta ani persi ge nyang ma te res da de ka bana ko persi ta ta do yo res.
Oops, wait. This is wrong. Something happened in the translation here. So there were some internet issues last night, and some of this is wrong. Okay. So this is okay. So the first mutation we're going to talk about is by substitution.
Da tangpo te nga su ge ka ta ge se na de ko ge chamji chi a chi go wa chi ge chab jen da chi ga chi go te ta ge ins.
This is where you substitute one of the nucleotides with a different nucleotide.
Da de ka chamji de ka su ka chi go la na nga su ge nyang de gu cha chi ge chab la ya nyang de cha jen da chi chi gres.
So in this case, we substituted the T with a C.
Da de ka se ba na gang la yo chi ge ti ti ge chab la ya si chi ge res.
This can cause the three different types of mutations that we talked about yesterday, or three of the different types of mutations we talked about yesterday.
Da de la te ne de khesang a su ga de she je sha ju ge ni ju ge riksum chi go wa ti che res.
First one is silent.
Tangpo de shin jam de wa.
Silent is we change that nucleotide, but it doesn't change the protein at all.
Te shin jam ge ni ju te na ka re se na ga su ge nyang de ge trip che te la ju wa tang res yin ba na ya tri la ju ru yo ma res.
The next one is the missense mutation. Missense is where you change one amino acid in the polypeptide chain. And the last one is a nonsense mutation. And that can either be a small change if it happens at the end of the protein, or a big change if it happens early in the protein. And we'll do a demonstration with this in a little bit. I apologize that I don't have pictures. With the internet being out, I couldn't access the pictures that I had from yesterday. Okay, the next type of mutation we're going to talk about is insertion. So instead of, so in the last one, we took this T and we changed it to something different. In this one, we're keeping the T, we're just putting something in front of it. We're putting another nucleotide in front of it.
That ngormagi pitsentena karichire ngasu that dena na pig tree tigi sabla ya shenda chichu barware mare. That tanda gi ninjun di kapsu karichigore sne tree ti ranjasha ne khoni gi tree tigi ndalola ya sarba chichu ko res shenda chichu ko res.
What type of, so this is what happens in the DNA code. What do we call that in the polypeptide chain?
That tanda di karina ni chungore sista bala ngasu ge DNA gitina chungorwa. Then dela tene de trisha juge ani trisig thongwa tigina la ngasu kari lab grace. This is a French shift.
Yes, great. Yep. Okay. And the opposite is going to be deletion. So this is what happens in the DNA code. We delete and remove one of the nucleotides.
Ta tige ngongi tenga su parola ya panjiche orwa panjiche res. Ta tige jela ya ku ane shendachi karo res stabala ku sumdon chigres parone maratemba.
And this also causes a
Then diletende te yande karchongres. Yeah, friendship. Yeah.
Yep, great. Both are the same. Excellent.
Kipares.
So, if you need to reorder your cutouts, if you need to change the order of your pieces of paper, go ahead and do that.
Ta tanda chisu panchi tisha yorwa. Pa riju tila ya ta ketin joa tangu yo pa na tanda joa tang roches.
Okay. Looks like most folks feel good about, well, show me with your thumbs. Feel good, need more time.
Dig science na laba dire churches tanda chuso ma gugundu sana ma dus churches.
Okay, good. Excellent. So in the order would go, the original is obviously the least changed, and then it would be the silent mutation.
Ta tanda nga su panu kada stigre sena tampote nga su la ya persistir wa re mare te la ya nyua nyinju du yo marwa te tampoche te jila ya ne nyinju shinjam nyinju ruju tres.
And then it would be the missense mutation.
Te jila ya ane nga su ton tu ase ton tu nyinju tres.
And then the nonsense mutation.
Te jila ya tome ge nyinju tres.
And then the two frame shift mutations.
Te jila ya ane nga su te karsa so
in general, deletion and insertion cause a bigger change than substitution.
Tatanda de kapsa chirki chana imbaina aneko kasa parula panji chiwa tang sumdon chiwa gi ninjuri orwa tege ane ngasuge dilaten de kasa kurga chap chiga chigorwa. I've
heard monks are very good at finding the one exception, though. So I challenge you to talk in your group and find the one exception where substitution is just as impactful as insertion or deletion.
Matamba yopati ane chikarsa chela ya kiba yoresa tenga tono chansonsrwa nestambala ya tanda dena ne ngasudela ya chamji chia chigorwa chamji chia nang chiti ane ngasuge karsa sumdon chiwa ta yana pranji chiwa chigorwa ti nangshingi ane ko nishik damba tinda chigores ta chamji kana la ngasu chamji kana ne ti orwa karsa sumdo chigorwa ta ti na ne kangi chi Ani sumdon chiwa ta pranji chiwa ta ta dawi ores tan koge shucheng chani baina tichi charo chidasa
2 minutes. 2 minutes to see if you can find the one exception. Do you think they understood what I was asking?
Yeah, I think so.
Because you're an amazing translator. Because you're such a good translator. A hint, this is not on your paper. You don't have a piece of paper for this. The type, you have the type of mutation, but you don't have an example of this. You don't have an example of it.
Kasa. So gala yo maris cheso pisa tesa tiga yo maris tata di imbana ngasu yinjur go regular tende tata kurwata.
You kind of do. You kind of have an example, but you kind of don't.
That pisa te chechen de lola yola mela do chires ta yo pachne re pachne res. Mare trivati marda. Trivati kutsu sa. Yeah, trivati rwa ta trivati nya. Ngasugo drumjiri but I'm just explaining. They probably didn't answer the question clearly, so I'm just explaining. Yeah, the one is friendship, right? Something is some of the one from the substitution is effective as the friendship.
Yeah, so friendships make big changes usually. There's one type of substitution that also makes a big change. And you know about that type, but there's a very specific criteria to make that type impactful.
Oh, now I know. I
think you know.
I know the answer.
Yeah,
now.
I know you know the answer. That way you can explain it good so they can.
Tarwa diri da. Ngasu ta todo shepa imbana rwa. Tanga sula drumjir yorwa drumjir. Ngime drumjir says to friendship. Friendship la ni yorwa. Samjik jiwa chita yana samdan che ni yorwa. Ta kugun jura shik chambu chitung yorwa. Re mare. Ta ti nanjing ki jura shik chambu tenye chi. Jenda ninjurang yores tada ngasuge dilaya samjik jiwan diginang la ya. Rwa. Samjik yasme yorwa. Diginang ke chiki ani drumjir nanjing ki jura shik chambu chik tengres. Tati kangere seshos. That was what I said.
Okay. 2 minutes. 2 minutes. So in what? Yeah, tell me more. Nonsense, but what has to? Why? What? There's a specific criteria. What about nonsense? Where does the nonsense? So if it's there, yes. So if nonsense is early, it chops everything off, and that's a really big change. Okay. When I bring it in 2 minutes, can I call on you and you can share? You can talk into here. You can share. Good. So I made up an activity for after this. I made up an activity after we talk about protein structure. I have no idea how it's going to go.
Yes, it's fine. It's fine.
It's an experiment.
Yeah.
Okay, I don't want this part to go too long.
Okay.
Okay, let's bring it back together. Okay, so would you all mind sharing what you thought? Or explaining what you came up with?
Nonsense.
amino acid laura de thawadala and gyoshu chimo thing gyoshu chimo kar thing do lana stop chart do those
nonsense with the stop code, so it makes a different like a big change.
Yes. And where does the stop code have to be to make a big change?
Ta tandagi tinado joya larwa ngasu chamshagi chida chorwa kun jroshu chambol hai la ya chamshagi dati coach lal hai guris.
Beginning.
Yes. Good. Beginning. Early. Yes. Yeah. Good. Okay.
Rwa tata de disarwa. Ngime drumjir tan dawu gushin chambu tete kalana tanda ngasu tome setris. Tome tigna la ya chamsha se ge dati on bain bana dikuyo maleswa. Ya chamsha se ge dati ku jro chambu tren la ya ane ku gut wala le gur mato ya jit wala ne jro chambu ting marwa gut wala guris.
Okay. So why are Miguel and I spending so much time on proteins and mutations? Don't actually, this is a rhetorical question, but why are Miguel and I spending so much time on mutations, but don't actually have them answer.
Ta game ge tangaran ge tanda ge ta ninjur tanga tzi kola ya chisto tina mabuchi karchine tango yores.
It's because the structure of proteins directly influences what they do in your body.
Tati karchine chisto mabutango no jinsen de kareses tambalaya ane ko ge tzi ge ko ge chako chigorwa. Tizigi chako tege ane kurang ge chele chigoro nga so zupuna na leka kare chigi ndu mdu sela telaya shukchen chembute gores.
DNA influences everything about us through the lens of proteins.
Ta nga so ge rigze tege ane tizi juli dete ane nga so ge kare chigi me chitelaya ane ko shukchen tege gores.
So proteins are what let us do all of the things that we do, whether it's digest food or grow new cells or think or breathe. All of those things are dependent on proteins.
Tata nga soge kasa chiga sinjuare utangare ane tila sopa ge zupu ge chegu dra chigorwa te chama tizila tani chongores.
And the structure of proteins is directly influenced by the order of amino acids and the shape of the protein.
Ta tanda ge koge tizigi kupa chigorwa kupa ti karigi tenke zugore sana amino churji ge pangram chigora ko pangram te ta ane kupa ti ge ko shukchen tegores.
And I misspoke. The shape is influenced by the order and what type of amino acid. So the different type of amino acid and the order that they come in determines the shape, which determines what that protein can do.
Tarwa tanda nga suge amino churji gi kasa amino churji kari yemen ta ne ko pangram kanda do chi dig yo metela ten de nga suge tzi gi chako tela yarwa tzi gi kopa tela shikchen tegres ne tzi gi kopa tela ten de ko gi chele la ya shite yores.
So we're going to take just a little bit of time talking about the different ways that proteins are shaped and the way they organize themselves, and then we're going to do an activity where we can see that at a large human-sized scale.
Tanda nga su de jela ya tzi ko pa tan tan do ku pang kan do chi dig yo me gi ti ne tan de shi ja gi ne ti jela ya ne nga su ge ta tzi ti chir tang de ta mi gi chi chong de na do ge nam ba gang la ya ani kanda do chi ko lang gu du ti la ya ani je gu chi do ya yes.
So there are 4 types of protein structures that we're going to talk about.
Da de la nga su de shi ja gi tzi gi chako yo ra ko pa ti ma do shi yes.
The first is called primary structure.
Oh
yeah, I forgot to mention, we are entering into our concept invention phase of this lesson, or the explain part of the lesson. So we're shifting from explore into explain or concept invention, depending on which model you're using. Primary structure just means the order that the polypeptides are in. So far, everything that we've looked at with translation is just primary structure. In order to understand this, you have to know just a little bit about how an amino acid is structured. And I'm just going to take 2 minutes to go over this very briefly. All of the amino acids that we're working with in translation have what's called an amino group with nitrogen, a central carbon, and a carboxylated carbon.
Ngasula diores tanda ngasugi tan deshe je shaju ge amino churji thamalaya ngasula ya ko la tripchala shisha ba bana tanda nga su amino gi rukha chi che ro amino rukha tle n yo sa chi che ani ko tiwa gi carbon yo sa chi si yo sa de che ni shenda tanda si ta o yo sa chi to wa de la ya carbon si rukha se gres di gi tripcha sam di ores.
All the amino acids that we're working with, this part is the same on all of them.
Da gala yo pi amino churji gi na ne amino churji sama gi na ne gala yo pi gi tripcha chi ora de sama ne chi par is samala.
The only part that is different is what is called the side chain off of the middle carbon right here.
Da tanda kusula manda gi cheperti kari ore se stambala ya chepre yo sati tanda ola art such doa tanda de nga chu ge surgi thawa yo ra surgi thawa de manda ores.
You don't have to memorize this, but you do need to know that there is an order to these three, this backbone. There's an order to this backbone because that will matter very soon.
Da tanda chesun de ngachcho tangu yo mare simba na ya gala ya tripi gi khurge ta backbone sana drum do chu chi ro te la ya ku param chi duwa o ti yang chi ngi gu gres karshila baina de yang
So when we look at the order of polypeptides, this is sort of what it might look like in what's called the ball and stick model. We start with the nitrogen, we go to the carbon that has the side chain, and then we go to the carbon that is double bonded to oxygen.
Chingwa chi orwa chingwa ga ramba kana do yo metila shisha de yores nandi bana tampo nga so amino research che and tigi jela ya ko tiwa ga carbon chi ora te che ani tigi jela ya nga so oxygen tan yam te tre da chu ge ani carbon ti ngores.
So that would be one polypeptide. Sorry, that would be one amino acid, excuse me.
The amino chirji chi res.
That would be another.
Amino chirji ni che.
That would be another.
And
so on and so forth.
I
want to draw your attention that there's a direction to this, that it goes nitrogen, side chain, double bonded to oxygen carbon. And that pattern repeats in this direction. In this This will become very important in secondary structure. Because secondary structure is the orientation of the polypeptide's backbone.
Tanda de kare sistambala ya kuge panram chdoa panram te ane kuge chakko niba ge ta ko chiga drum do chagres.
And we're starting to get into more complicated arrangements.
Ta tanda de ge jela ya ane nga su tele ge nyokta chao ge ane ngu chilan chagres.
There are 3 types of secondary structure that we're going to cover, although there are many more in actuality. They are parallel, anti-parallel, and helix, and we'll go over them all in more detail. You don't need to memorize these, but it's important for you to know that there are different levels of organization in a protein. So don't feel like you need to memorize exactly what these are, but understand what level of spatial organization we're talking about. So this is our friend, the amino acid. Just to review, right? Nitrogen, carbon, carbon. So this amino acid, first let's talk about parallel structure. Notice that this polypeptide goes nitrogen, carbon, carbon, nitrogen, carbon, carbon. if that orients next to another part of the same backbone in the same direction, we call that parallel.
That tinda do chi jenda chi ko nitrogen carbon le ro akhorong geta chakko go chu ti tinda do nyam te yo pa bana shok chila khasok chila yo pa bana ne dela nga su ge nyam shi grace.
There's not a great example of that in this drawing, but you could know that this strand is going to be parallel to this strand because they are both going in that direction.
Da de kap se nga su dela ta ba bana ko ge phajo ga da dela ya ko de chok chok ma bu do phajo ke ko pa de ta phajo ko pa de nga su ge khaso ten dus ro re mare de ta ane dela yo chu din sha yan ko nyam shi cha ge yo re san cha ma ru go yo wa.
The next one is anti-parallel, and it's just the opposite. It's going in the opposite direction.
Ta de ka san nyamshi ba ka sha dog de ju ro nyamshi dog pa de res ta de ma na ko shi phan ro chi char du yo res.
The reason we're talking about these is depending on if they're parallel or anti-parallel, the polypeptide chain will take on slightly different shapes.
Ta de nga su ge nyamshi ba ta ya na dog pi nyamshi ba de la te ne de te nga su la ya titsig chwa chi ro te la ya yan ko zu ta man ro jing res.
This part right here and this part right here would be anti-parallel. Because the directionality of the chain is going in this direction here, and then if you follow it around, it's going in this direction here. So they are going in opposite directions. Okay. And then the last one is a helix, and that's just this right here, when it's going in a spiral around itself. Okay, two more. Tertiary structure is how different amino acids interact with each other based on their side chain.
Nibati saro ta sambati bayana ko chako sambati bayana ko ge ta amino chudzi mandra ge ko khori ta nyamte dejor kana ro chi chi metela ti res.
That's shown in this diagram here with these side chains interacting with each other in different ways.
Ta da printing na ta sa yo ta ding na yo pi ge ko ge surga thawati khusu phansula ya dejor ka ti chi ge opati ta yo res.
And these are important because they create big loops and folds in proteins.
The kitchen which are gores, Kashila Bambana, Kuta, Chocho Chayata, Chujata, Tigi Nitanting Gores, Titsig.
And the last one is quaternary structure.
That Dikares is Tambalek, Chako Shiba de res.
And this is how different polypeptide chains come and interact with each other.
That Dikares is Tambalaya, and a Titsig Mandawati, Kusu Mandawa Parolaya, Dejokana Uchungu Patiris.
An example of this is hemoglobin. Which is the, sorry.
Yes, please.
Oh, which is the protein in your blood that helps carry oxygen. And it's made up of 4 separate polypeptide chains. Okay. We're going to move into the application phase. What we're going to do is we are going to turn all of you into a protein. And we're going to look for the different structures within the protein that you create. The way this is going to work, where did my ropes go? Here they are. I haven't tried this before, so we're going to see how it goes. You're going to be in, your tables are going to group into 3 tables. So this will be one group, that will be one group, and the back row, you will be one group. What we're We're going to need to have 15 people holding each rope.
아하 다단다 데라야 쿠파시도 탁바로시도 데라야 아니 미 중앙 근주고요레스.
And you're going to need to stay in order.
아하 네 제초 미 중앙지 아니 제초 방름 따도 되고요레스.
And you can't let go of the rope.
아하 네 제랑게 타바데 르넨디그 마레스.
If there are more than 15 people, I'm sorry, but if graduate students, if you wouldn't mind stepping out, that would help keep the numbers even.
아하 타케테 미 중앙에 마오요 바이바나 세리게야 칠롤 텀바이바나 네 젠다 코술란 주시제 코케라그와스.
And hopefully you can sit back and chuckle with the rest of us as we watch this happen.
아하 아니 제초 파칠롤 텐데 땅가 소기 간다도 지지게 요메티강라야 아니 따토체다야 지니 케무게나 드고레스.
So, for example, I'm going to number you all off, and then the other the other two groups, you can number yourself off. So you'll be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15.
아하 다 방르데 테스와 탐보데 코슬라 탐보니 정가 바다네 젠다 수수수게 땅가테 수수 테산디레스.
The way that you're all going to hold, sorry.
Yes.
Oh yeah, I talked for a long time. Sorry.
No. I'm just asking them to give them numbers by themselves. Yeah, you said that.
So 1 will hold here, 2, 3, 4, 5, 6, so on and so forth. Yes?
Okay.
Let's so you're numbered. Let's have you all number off. And then you all number off. Once you know your number, we can walk to the sides of the room. So this group, let's have you be over here. Middle group, let's have you be in the back of the room. Back group, let's have you be over there. Do you all have your numbers? Here's your rope. This is all the rope I have. You have to switch. Yeah. It's all the rope we had. Okay.
Kare da thoba the ta de cheme lapa sigri jina de ko sa lapa chik jina de gre sa
Yeah. Okay. We're going to change the instructions. We're just going to have them hold hands. We're just going to have them hold hands in order. So instead of they're going to let go of the rope, they're going to like hold hands in a chain.
Okay. Yeah, that tabodi do so wa tanda de ga su de tabade la ba ji de wa that tabade le ro ji sa e tabade le nu ji la ga ri che re system ba la ya ani tanga tanga te so tanga te pa na se param ji ani la ba ji ro ji sa thaba le ji la ba ji ro ji sa tampo nya samba sho ga tu pa tamba je pa la ba ji ro ji sa thaba le sa.
It's too short. It's not going to work. So you're going to have to, hold hands in order. You can spread out now. You can spread out. Spread out. I can take the rope. Oh, he's got the rope. Thank you. Okay, good. But in order. So yeah, good. Okay, good.
And the problem kanda yoba lapan jiro chesa tamponya sambasya tampo kware tampo dire
Don't cross your hands. Don't cross your hands. That will be complicated. Don't cross your hands. Don't be too hard. So just like, yeah. Just like this. Good. Just like that. Just like that. Good. Good. Great. Okay. Where is Dawa? Okay. No, don't join. Don't join yet. Don't join with the other group yet.
Yeah,
okay, good.
Stand. Yeah.
Okay. Okay. Does everybody know their number? And you are in order along your chain. Okay. Good. I'm going to show on the screen. I'm going to show on the screen all of your numbers, and your number will have a shape by it. Once you know your shape, you have to find somebody else in the polypeptide chain that shares your shape, and you need to be close to that person.
In like each peptide or all together?
Yeah, so let's say two people have a triangle. Let's say number 15 and number 2 have a triangle. Those people need to come together and be close to each other.
Okay. In each peptide, right?
Yes.
No, not all together.
Not all together. Each group, yes, in its own group.
Okay.
Yeah. Good question. Thank you.
Ni gelage ya la tangka chitegre tangka telaya ane tangka telaya zota chiores picha shapaina tatsimo mashe tangka chilaya tangka roa tangka chunga ta tangka nipa nichalaya konga zota te susma chiyo paimana kat chigure sena tangka nipa te ta tangka chunga chiyo roa o ten nichata thanyu legures.
But you can't let go of the hands. You have to keep your hands together. Okay. Let's see what happens. So here's what you are. You might be a triangle, a circle, a cross, a square, a star. If whatever you are, you need to find somebody else, everybody else that is your thing, and you need to be close to them.
Taro cheese roa. Tangka tampani chunga parta zuta dua. Roa. Lakpa le yo mares roa. Ta kari chiwa lakpa malu igane susu ga zuta kari du roa. Ib kari rindu tigi tita thanyu logugres.
Okay, good luck.
Oh yeah, ta tashi dele sa ta zuro na sa.
Yeah? Good. Who's the lightning bolt? Who's the lightning bolt? Who's the lightning bolt? 13. Who's number 13?
They're parallel to max 1, 30 and 6.
What's that?
I mean that each of them we have. I'm
You
13,
good?
13 to.
Yeah, 13, so you don't have a match.
Yeah, I don't know.
You can kind of go wherever. Yeah.
Pretty right.
But you need to stay attached. Yeah. Okay. Is there anybody that didn't find their group? Everybody found their group? Okay. Is there anybody that did not find their group? You're the lightning. But everybody else? Okay, good. Is there anybody that did not find their group?
They find the group, but this space is a little bit congested.
Yeah.
So if there's more space,
It's supposed to, this is part of it, is to show that it's like tight and compact. This long thing is crumpled. That's part of the point.
Okay, good. Okay.
Yeah. Okay, so this is going to be our non-mutated protein. This is our original protein.
That the Tandangasu Gala Tesha Jutirwa. Nye Kayam, but that is like that. The Dicare system Balayanga the Pishiti is a Pishasatra, Dicaresena, Nye. Did and then you to make you to the
So look around, notice where you are relative to all the other amino acids before we mutate.
Tatanda and Susugi Damlaya, amino chizi kare, that's what the kare into Chituna Chichirochis, Rata Tuna Cheti, Tandangasuta, Ninjur Matangela Kandanoshin to the Tuna Chirochis.
Don't move yet.
Tando Kushamata.
So we have a silent mutation. So we changed our DNA code. Do we have any change in the protein?
No.
Okay. Next one. We have a substitution. So, number 3, you used to be a yellow cross. You are now a green moon. Okay, so number 3, find your new home. Find your new home, number 3. And everybody else needs to sort of move around to help that happen.
That's somebody that and then somebody to Korangi, that Tiladura, Chushita, Tanyu Duwa, that Tanyu Duala, and that's someone Gusheta that could have a Chirochisa. Yes.
Okay, back.
This guy.
That was fast. Good? Okay. Looks pretty similar or very different? Pretty similar. Exactly the same? Yours is exactly the same? Okay, so we have some that are a little different, some that are exactly the same. Do we think this has a big impact on biological function? Okay. Next one. Don't worry about the original. So the next one is this. There are many things that are different. So this is an insertion mutation where we frame shifted. Everything shifted. Find your new home. Find your new home.
Also, they need to change themselves as well, right?
Yeah, but they keep the same order. They have to keep the same order. Yeah. It's okay. Keep your hands together, but you can unravel and come back in. But you need to keep your hands together. Already rearranged? No. You're already okay. Very cool. Different or not different?
Very different.
Very different. Okay. Already good? Okay, good.
Hello.
Good? What's the next one? Original protein. Okay. Good?
Yeah.
Okay, sir. So this was an insertion frame shift mutation.
Aha, tadadirwa. Ngaso ngime drumjigana parola perjichiatres.
What do we think this had a big or a small impact on biological function?
Aha, that dilated the ngaso chingi cheli telaya suction chambu your survey, chonchon your surveys.
Bigger. Okay, good. Next one. So again, just remember, don't go back to this, but just remember our original kind of, no, don't move. In your mind, remember what the arrangement of the original protein was. Our next one is going to be a substitution nonsense mutation.
What
happened? We stopped. Yes. So 13, 14, 15, you're gone. Okay. Big or small or medium change?
Chepe karidu sonsa, juruati chembu teswe, tis teswe, nyon tesonsa.
Small? Some people say, I hear some people say small, some people say medium.
Yeah, kashigin jura chembu, chun teson lagudus, kashigi.
Okay. Our last one.
The tamati is
another substitution, nonsense mutation, but in a different place. 5 and up, you're gone.
Okay.
What impact did this have on biological function? Big change. Yes. Okay. Oh, go back to your original protein. Go back to the original protein. It's up here. So remake your original protein. Fold back up. Okay. Okay, so far we've been changing our primary, secondary, and tertiary structure. So far we've been changing our primary, secondary, tertiary structure of proteins. What structure have we not done yet?
Chako tamponya sembasi desija songa tangirwa. Chako tamponya sembasi desija songa shiva desija sa. Tenani tamponya sembagi juruan dua itu nggak sudah desija baris. Ra. Tapi tenani desija major kali hadus. Yeah, the tertiary.
Quaternary. This.
Oh yeah. Quaternary.
Okay, good. Okay, so we have 3 different polypeptides around the room. This symbol is very attractive to the other versions of that symbol. So all of the yellow crosses from the 3 different polypeptides, you need to all be close together.
tang serbu janggu te wa tang serbu janggu te tang ka san tang de da ti yu ji chama ne thani ulan ro chis.
So we need to join our 3 polypeptides in the back of the room and have our yellow crosses close together.
Da ma do re sa ma do de tang ane tang serbu ba tu nya thani u de re sa jen da tanyam ta. Hello marda ta de ti do marda.
Yellow?
The number for this color.
Oh, the number. Yeah. 3 and 4.
Yeah, number simbata shiba.
3 and 4. Yes. But everybody else needs to stay attracted to the people that you were attracted to in the original protein.
Ra tandage chechugo zuju chi wa tele jondo yo mara. Jondo yo mares ra taraja de. ane tad din cha mando jablando de ta phajok ke sambata shiba gi dam la yan de gi sambata shiba do go do wa ta peps ta tre ya do gore ta do gore ta do ta do
The purples 5, 9, 10. Excellent. Good. Okay. Oh, I can't reach. Okay. Oh, we just had a different type of mutation. Purple squares, which is 5, 9, and 10. Now you all are attracted to each other. Okay. Okay. Very good. All right. Thank you. You can all go grab a seat.
Oh, teaching us.
Okay, so in your journal, we want to take 5 minutes. Sorry, go back. We're now in the reflection part of the learning cycle, or the 5E version of reflection is evaluate, I think. Evaluate is the last one. For 5Es. Yeah. Evaluate. So we're in evaluate or reflection, and I want you to write in your journal your reflections on these 2 questions.
Da tanda nga so de la ya samshi che I nga deng tina na nga so gidir wa da chenje gi thamater wa tel de ores da tanda su su gen detep gna la ya tuani do de ko la ya ne land roches kama nga gren la.
The 2 questions, yeah, thank you. Yeah, very good, very good. First question, what impact did changing the primary structure have on changing the other structures?
Tua tangbuti ka reses tambala nga su ge tzi gi ko pa la ya ko ramba shine chi duwa. Ko pa tangbo de la ya, tzi gi ko pa chakko tangbo de la ya jwa chimba de ge ane ko pa jenda la ya shkuchen ka rezu gres.
And how might that change the function of the protein?
Tin jwa de la tena deta ne tzi gi jeli la ya ane shkuchen ka tegres.
So 5 minutes to write on that, and then we will go on to the next thing.
Ta kama nga grang la te ko roches, ante jela nga so jenda ngutu che yes. Twa go tu sa. Twa te tangbo te I'm just repeating the question.
Yeah.
Dua tambuti kala na ngasuge tizigi ta ko chokola tambana choko rimba shi desi jason ro tamponya simba jiro jason rimba tambuti la ya ko jiro chi thepa imbai na ani tigi choko jenda yo ta nyo simba shiba rimba ya dugdu wa remare tat tinya tapsu ja tong mando wa tambuti la jiro chi batile ne ko choko jenda tuzla jiro kari tegres yuchen kari tegres ro tambuti la tilatene ta tambuti la jiro timba tilatene ta ani tizigi jeli la ya yuchen kari tegres jiro kari tangres yuchen kari tegres.
Okay. Hopefully, you are starting to get fairly comfortable with the central dogma idea that Miguel explained yesterday.
Aha, da chitsula ge Miguel kesang oto chijuge tawanyang puchi orwa pita kanda do chuba da chaji de ser duya gerwa yes.
We have DNA goes to RNA, goes to protein, goes to traits.
Aha, tate tabai bana ga su DNA ne RNA je RNA ne tzi ne ga su ga cheshi ti ngoyong go res.
And if you change or have a mutation in the DNA, you can sometimes have a change in traits.
Aha, ta DNA la ya ninjula tene jiro chuchong bai bana ya sam la ya ga su ga zupu gi cheshi la ya jiro du res.
And different types of mutations cause different different types of mutations cause bigger or smaller differences in traits.
Aha, ta ninjur ge rig mando la tene de tenga su cheshi nang ge chechi jitok mando ta ko de la ya ji jitok la ya shi chilo ji ro o te teng res.
Okay. Yes, no, still kind of fuzzy.
Is that?
Okay. Everybody's got to put a thumb up or down.
Okay.
Okay. Most thumbs up. If you are still confused, that's okay. Let's talk at lunch. Okay. We're going to switch gears. So we've just gone through one whole cycle of the learning cycle. Yes?
We're going
to go through it again, but with a slightly different topic. I'm not going to go through Invitation to Learn this time. I'm actually going to skip it because I feel like when I look at my class, you are already engaged in learning.
So
I didn't label this here, but this would be Explore. We'd be back in the Explorer session. So we've gone over the central dogma of how we go from DNA to traits. I want us to get to a place where we're thinking about how mutations impact the diversity of life. To get there, I think it would be helpful to spend a little time reviewing genetics. And more specifically, heredity. And we're going to do this very quickly and as a practice quiz. What is a practice quiz? Thank you. Okay, let's try. So I did this last night. I'm sorry that this is not in Tibetan. An area on a strand of DNA that codes for a given trait is called a what?
Kasa di ngasuki chasing ijen jike da duper shebi roa ane DNA ge trapcha tila karse grace chashitela
10. Yeah. Gene. Yes. Excellent. If you knew that, give yourself 1 point in your mind.
That is rigid, right? Rigid say lengthy single panasus, lapanasus,
We often draw these as stripes on a chromosome. So this would be a gene, this would be a different gene, this would be a different gene.
That thing I took it, remove on the chance, Tambaraya, I took Chizik Ganlaya, and it's Chontamanda, Chitan Chitomachigores. That could get Chontamando, get to chat and it's most race.
I realize as I look at this that this is a bad representation because these are different copies of the same chromosome and the genes do not line up. So this is actually a bad drawing. Sister chromosomes should be identical. These are not. But I digress. Oh, that's impossible to see. Oh, sorry. But you can just go with the words. Because we have 2 copies of each chromosome, we have blank copies of each gene.
Tatanda Natsulaya, any Kasa, Twizitim Bana, Twizitilaya, any Ko Kasugre, Ku Persinjiri or Wa, Persinjiri Stambalaya, and then Natsuga, Rizila Chambana, Rizigi, any Persu Katsoyores, Rizila Persu Katsoyores.
Talk for 10 seconds to the people at your table.
Kasachuganala, Chitu to the Jessa.
1,000, 2,000, 4,000, 5,000, 6, 7, 8, 9, 10. Okay, how many copies?
Copy Resa.
2. Excellent. Yes.
Yes.
So these actually show up pretty good on my computer. I don't know why they don't show up on the projector.
But this is the Lolega Yabutongo to say, they tend to buy Yabutongo Karsina Hakumasons.
Yeah. Different versions of the same gene are called what? 10 seconds to talk to your table.
Tanda Riziki Pacherwa, Riziki Yankuga, number Riziki Palaya number Mandoyorwa, or Tila Kalagres.
1,000, 2,000. Oh, they just shouted it out. 10 seconds to talk. 6, 7, 8,
Go.
9.
2, Sas.
It's allele. Yes, alleles. Different copies of the same gene are called alleles. So you might have a gene that controls for hair color, but there are different alleles that will code for different colors of hair.
Stragi Centralaya Tangzin Chinigi, Rigzichi or Remare, Rigzichi or Pakashila, Yantiku, Rigziti Tangzin Chiagi, Nabatela Yangasu, a number mando, Rizila Nabamando, it's not Elimando Yores.
So is the answer right here already?
No.
Okay, good. Okay. Different combinations of alleles create a variety of different what? Blank. Talk for 10 seconds. Talk for 10 seconds.
Tadikala Narwa, Natsuga Elili Mandawa, Yamsichi Balatenetis, Ra, and Mandawa Courage Mandawa Yongres.
6, 7. Traits. One number. Yes, very good.
La.
You must have had very good genetics and heredity teachers. Okay. Mutations can further increase this variety or even create blank types of traits. Mutations. Go ahead.
kugi ta natsuko rangjan sekura natsuko rangjan di manta tangwa ta yana ani ta chi tsachidwa ta cheshin di ani tumbar chigres kari cheshi tumbar chigres
10 seconds 2 3 4 5 6 7 8 9 10 okay different or new traits yes excellent
what
different or new
okay
yeah
cheshi di manda ta yana cheshi sarva tumbar chigres
good okay different works as well
manda wala na digdus
okay only one or two more because DNA is passed from parent to offspring alleles and the traits that they code for are passed from parent to blank
Radanga so data DNA thing as a permanent pugila to start makore jutuchigo resra this time la yang as allele ta ni cheshichigo ta ko allele the trace they code for ta ko tanda DNA ten de tan ko allele ta trait thing gorwa ti ani permanent selected res offspring
good yes sorry to put you on the spot with
no it's good yeah it's good
okay I think this is the last one if we know what alleles the parents have we can predict what alleles and traits the blank will have
the cat is ngasuge phamala yobigirwa ani allele te karim bahaku yobaina ani ngasuge ta suchila yopige allele ani chopochit kroes yeah offspring
yes offspring excellent for extra credit what do we call this thing Mendel cross yeah Mendel cross or there's another word for it Punnett square nice excellent okay so it's pretty close to lunch probably should not we're going to pause here so when we come back from lunch here's what we're going to do we're going to look at how mutations change the frequency of traits in a population over time.
Ane chiga ta tin rampu chikrang la ya ane ko chondi chikrang la chongbe ge cheshi chigorwa cheshi data frequency system bala monglo chongya de sete la yan jiro kanda ru chitang yo metela tagins.
So I just did a quick assessment in like a practice quiz style where I tried to understand where you were with your understanding of heredity.
Ta tandangi chesula ya de gala ya ngasu tan jobo tse do tangatila lenja bala tene dete ani chesuge kasa ngasu junjingi namsha chigora junjingi tatuti kola chesu katso chi shingu yo meteta ba yes.
It was clear to me that many of you have a fairly good understanding of heredity, and if you didn't, that's why I was trying to have you talk to the people at your table so that people that maybe weren't as comfortable could still hear your ideas.
Ta tanda dilatene deta chesu ma chishuge tanga tsuke junjin chigora jutu chia chigora rgzi jutu chia ge tegu ko ge chi tatsu tsu ma chi singdus pe abu singdus ma chishu ti singdus ta na shenda ga suge chi karsa toxic gane chesu yam te yang tutu chi ge chi te imbaina ya chi machine te ge jenda te ge chi ge tatsu kana ru chi chi ko ge te ko ka tribayens.
So that is a way you can both assess and teach at the same time. So now we have, as a class, we have a basic understanding of heredity enough so that we can move on and talk about mutations and how they change traits over time. Okay. I think it makes sense to stop here as opposed to starting something and having to end in 6 minutes. So we're going to break here. I will see you all here at 3 o'clock, and we will finish up this afternoon.
Ta tanda de chinggu zi sha sha ba na de ya gom du stis ta malaya tanda kama khasichi du da ba na person tang is so sumba gan in ta balaya ane sumba gan face ta balanga so George Saba de gan la ya ane ngoto ji is.
Thank you all for a great morning. See you in a little bit.
Hoya tu chinas.